Pedro Mediano is an assistant professor in computer science in Imperial College London, and previously he was a postdoc in the Department of Psychology at the University of Cambridge. His work focuses on the mathematical principles of neural information processing with applications in consciousness neuroscience. So I will give the stage now to Pedro.
Amazing. Thanks a lot, Joel. Thanks a lot for everyone for coming here. As Joel mentioned, my background is perhaps a bit different. I come from a physics background, mathematics of complex systems, computer science, lots of applications in psychology and neuroscience. But I'm gonna kind of turn a bit things. I'm gonna turn the symposium to perhaps a bit more speculative angle. I'm not going to be presenting any new results, but I'm hopefully going to give a bit of context in terms of how we want to move this whole research program forward. My talk is about measuring consciousness with perturbational complexity. So before I start on talking about the complexity itself, I wanted to just say a few words about measuring consciousness. Why is that something that we could or we should want to do? What I want to say is that there's actually it's a problem that has both a response to a clinical need, but also adds a lot of scientific value. So measuring consciousness will indeed have lots of clinical and scientific implications. On the clinical side, for example, we know that there are patients that can, during a sort of disorder of consciousness, they can, for example, result in a locked-in state. So patients that are behaviorally unresponsive, and yet we strongly believe that they are conscious. So having a sort of measure of consciousness with that report would be tremendously useful for treatment decisions, for example. It could also be very useful for, say, anesthesia monitoring and regulating doses of anesthesia and the like. And then what I want to argue, and what I think of, some of the spirits of all of us here in this kind of line of work, is we want to say that if we develop a kind of piece of, science or equipment or algorithms that can address some questions, we can then bring it back to the more basic science and ask questions about the emergence and the development of consciousness. So the research question here is, can we measure consciousness in the fetal brain, and then what can we learn about consciousness more generally in the process? So the take-home message that I want to kind of give you during the next 10 or so minutes is that there is indeed existing theoretical and experimental basis for measuring consciousness, and that there are some possible, at least conceivable applications to infants and applications in utero, although some changes to standard measures are required, and that we have a bit of a roadmap for how we could go about getting these things validated. So let me start talking about information theory and consciousness. Not very much, but just to give you a bit of an introduction. So when one talks about theories of consciousness, of course, there's quite a few things available in the market. One of them that I am kind of like is this thing called integrated information theory, proposed originally by Giulio Tononi some time ago, but that lots of people have worked on it ever since. And kind of the IIT is basically this ambitious theory of consciousness that aims to develop this quantitative measure of consciousness, typically denoted by the symbol phi or some version of it in case you're familiar with it. So the idea of this is that you can somehow get a number that represents the kind of overall kind of coarse-grained level of consciousness of a system. And so the key premise of IIT is that a system is conscious if it is somehow more than the sum of its parts, right? So if the different parts of the brain interact in a way that is irreducible, defined in some mathematically precise way, then we can say that the system is conscious. And in its sort of strongest form, IIT is based on these sort of 5 axioms that consciousness exists and it has some compositional structure and whatnot, and then you kind of turn a really big crank and you end up with some of these 5 measures. And IIT has lots of good and bad things. One thing that is probably bad is that testing this thing empirically is actually really, really difficult. So one thing we've been thinking about for a few years is sort of what changes can we do to IIT, or what requirements do we need to relax so we can use it in practice. And then we kind of developed this concept of a sort of weak form of IIT, which is basically an empirically driven framework to test specific components of this kind of broader theory of consciousness that is IIT. So we grabbed some ideas from it. So 2 of those kind of 5 axioms that you saw earlier, 2 of those 5 little bubbles that are particularly amenable to experimental study, is the idea that conscious experience is informative and integrated. Now what do we mean by that? By informative, I mean that any given conscious experience is informative because it is very different from a very wide kind of possible range of other conscious experiences, and it's integrated because we don't perceive reality in patches, we perceive that sort of integrated whole. And for example, philosopher Tim Bain, that has, we mentioned earlier, also has some of the work on, say, the unity of consciousness and related phenomena. So how do we measure these in practice? One method that has been developed a lot, which is the sort of perturbational approaches to measuring information integration, proceeds by basically, say if you have a system, you perturb, you make a sort of causally independent perturbation to some of the elements. And then kind of in a somewhat cartoonish way, we can imagine a spectrum of things happening. So if you have a system that is sort of disconnected, and is sort of perhaps locally interacting in pockets but that don't communicate globally, then what would happen is if you zap one of these guys, then you will have a local ripple of activity and it will die out, and you will get this sort of relatively short-lived burst of activity. On the other end of the spectrum, when all of the elements of the system are very strongly coupled to each other uniformly, then if you perturb one of the elements, then you get this sort of very strong, kind of long-lasting and uniform response. And it's only somewhere in the middle that if you perturb one, then you'll get this sort of waves and ripples of reverberating activity that propagate in a very complex-looking way across the whole system. And it's indeed in this kind of in-between the loss of information and loss of integration that we believe consciousness happens. So how is this operationalized? This was work led mostly by the group of Marcello Massimini in Italy. And they developed this thing called the Perturbational Complexity Index, which is really, really good. Now there's going to be a bit of a busy figure, but let me walk you through it. So this PCI was proposed by Alan Al-Qasali in 2013. And the way it works is basically you have, typically in the standard paradigm, in adults, you have an adult subject with a high density EEG cap and a TMS stimulator. So at a given time, you stimulate with TMS, and you see the sort of ripples of activity that propagate all across the EEG, and you can see sort of the topographical distributions here. So you can see that really kind of the focus of activity does move around, reverberate, and change. Then what you do is you can kind of you can do a source reconstruction to get a representation of this activity across the whole cortex. You binarize this, so you get basically a brain map of zeros and ones over time, and you get all of this thing into a really big matrix. And that matrix, kind of what the idea that they wanted to sort of capture is, well, how complex is this matrix, right? Do I get just a big burst of activity that dies out? Do I get nothing at all? Or do I get this sort of really funky interplay that we were seeing in the previous slide? And the way they did that is with an algorithm that's called Lempel-Ziv compression. And that's what we're going to look at next. So let's look at Lempel-Ziv complexity. There's a measure that comes from the engineering literature back in the 80s, but it's been repurposed and it's now increasingly popular in neuroscience. So it's a measure of effectively complexity for time series data. And loosely speaking, what Lempel-Ziv does is basically it counts the number of different patterns that you see in a signal. Right? So for example, if I have a really funky signal composed of two symbols, A and B, imagine that, for example, in EEG could be A happens when the signal is above the average and B happens when the signal is below the average. That's A and B. I haven't actually thought about this. But yeah, so you have this sort of very funky signals of A's and B's, and then what you do is we're going to sort of carefully cut this signal in different patterns, and you might get something like this. And then you count the total number of patterns. Then if we have a very simple signal like that, you do the same thing, you cut it into different patterns, and what Lempel and Ziv showed back in the 70s actually is that for complex signals, the resulting number of patterns will grow larger and grow faster than it does for a very simple signal. So that, for example, the signal on top would be an example of high Lempel-Ziv, the signal below would be an example of low Lempel-Ziv, and this, for example, what you might observe if you apply this thing to say REM sleep versus non-REM sleep, as an example. And kind of mathematically, this Lempel-Ziv complexity corresponds to a statistical measure known as entropy, that again captures this idea of a system with high entropy, visits many possible states, and kind of goes through many different configurations over time. Where a system with low entropy is sort of rigid, robust, doesn't explore its kind of dynamical repertoire very much. So how does this actually work? So how well does this actually do? Very well. So I'm going to show you some experimental results again from Massimini's group. This thing is extremely good at discriminating between states of consciousness. So here we have about 30-something subjects, both in sleep and wakeful rest and in sleep, and also with different anesthetic agents. And you can see that basically you can just draw a line between the conscious subjects and the unconscious subjects. And that's regardless of the intensity of the TMS pulse, it's regardless of where exactly in the brain you stimulate, regardless of which specific anesthetic or whether they're asleep or awake. The really good property of this thing is extremely robust. There's an absolute value that generalizes extremely well across subjects and tells us a sort of absolute measure of the overall level of consciousness. Again, all of this in adults, I would say, to be particularly clear. This also applies to patients with disorders of consciousness, where you see this sort of very, very nice kind of gradual increase from patients in an unresponsive wakefulness syndrome all the way back to patients in a locked-in syndrome that are basically indistinguishable from healthy subjects during wakeful rest. Cool, so far we've talked a little bit about PCI. In the 2 minutes that I have left, I'm gonna kind of rush through kind of the challenges and opportunities that we kind of foresee for this type of procedure to be applied in fetuses and infants. So the problem with PCI is twofold. One is that it involves a TMS pulse, which is of course fine in most adults, but not really applicable in infants at all. And it also requires a high density EEG recordings, which is also not currently feasible in fetal MEG. For the second one, there are algorithmic and sort of equipment improvements that are happening. I'm going to focus on the first one in particular. So how can we come up with a feasible alternative to this TMS pulse? And to do this, we're going to kind of bring in some ideas from the framework of predictive coding. So predictive coding says that brain regions, basically what they're trying to do is going to predict their own afferent signals, so what's going to come to me in the future. And that neuroactivity basically reflects these failures of prediction, so these prediction errors. And you have this sort of hierarchy that goes from the sensory periphery upwards in some sense, and that you have these prediction errors and these predictions going up and down the brain. So the idea that we had was basically, well, can we use these prediction errors that are sort of endogenous, internally generated signals to simulate this sort of endogenous perturbation that is sort of like the TMS but not quite. To do this, to induce these prediction errors, then that is where kind of the local global paradigm that Catherine explains comes up, right? So we can use this paradigm to create an expectation via basically priming the subjects with a standard tone, and then we violate this expectation with a deviant tone. And again, as Catherine showed, we have this sort of very strong effects. This is also for adults, around 300-ish milliseconds post-stimulus. And we can, the hope is that we can use this signal and look at the ripples it generates across the brain, compute the impulsive, and get a sort of analog of this PCI index. So this is sort of the overall pipeline of this sensory perturbational complexity, or SPCI, as we have dubbed it, which basically means, well, we can stimulate the subject with some tones, or a sequence of tones with this sort of local global structure. Then this would, in principle, if kind of all of the ideas of IIT hold, this would elicit this sort of dynamic interplay of activity across the brain. We can grab the E or the EMG signals, compress them with this impulsive algorithm, and then get a measure, an estimate of this sensory PCI or SPCI measure. That's the overall pipeline that we have envisioned. Of course, there's still a long way to go, so there's a roadmap to validation. What we want to do is first start with adults, validate this sort of sensory paradigm in adults, validate its sort of translatability from EEG to MEG, then move down to children, and we find basically one thing that we really need to do is kind of find the optimal parameters for this sort of sensory perturbation that result in an adequate signal for later compression, and then mitigate the effects of this low SNR. So try to find algorithms that don't depend on having such a strong spatial resolution, and then we can move on to infants. And then we can, for example, contrast against the arousal data, such as the sort of hardware variability that Catherine wants to present. So with that, I'm going to wrap up. I'm already slightly over time, but what I want to say is that perturbational complexity is extremely useful theory-inspired measure of consciousness. It has been thoroughly tested across clinical conditions in adults, and we, despite the fact that we need to, find a TMS alternative for fetuses and infants, we do have a validation program in place. And with this, I'll open for questions. Thank you very much.
yes. I maybe I have two questions. One short one is actually, it is some such like the idea is instead of using the magnetic TMS, I just introduce something implementing the brain and then use this as a perturbation to do the same works.
Yeah.
Okay, this actually basic idea. So the second thing is like, I see you use a sequence, right? So even you use auditory, something like that, it's really relatively low frequency, right? So my question is, what about if we use something like SSVEP, such a really high oscillations as a stimuli? Will be getting more robust SNR? So because we will know if we use SSVEP or such like that, such things, we can get very strong or local perturbation of the signals. And I just want to hear your ideas. So if it is possible, so I just use SSVEP data on your method. So
yeah, I mean it does seem plausible. I mean the only reason we focused on this kind of local global paradigm is because these prediction errors are sort of thought to be endogenously generated, and that is nice because if you just have some kind of sensory perturbation directly, then that is processed by the whole kind of cortical and subcortical machinery and everything. So we're looking for something else. But yeah, I'm sure that other approaches are perfectly worth trying.
Time for maybe one more quick question. Anybody? Okay, if not, then I will take the stage. Alright, so thank you very much, Pedro. Okay, so I've already introduced myself. I'm here to talk to you about how we apply some of these ideas that Pedro just gave us to actual empirical data that have been recorded here at Tübingen. All right. So you just saw this figure. You have the roadmap. You know why we're interested in the entropy of neural responses to sensory perturbations. So remember, the SPCI is a spatial temporal pattern in response to a perturbation. And what I'm going to show today is getting close to this, but not quite all the way there, okay? So we have the temporal dimension of this activity, but for reasons that I'll explain in a moment, we don't have the spatial dimension. So it's not quite the SPCI, but we could say it's an approach that's inspired by perturbational complexity. So we applied a few different entropy measures to our data. I'll just very briefly skim over them. So the first are these signal compressibility measures. Pedro already did a great job explaining this. It includes Limpel-Ziv, which he mentioned, and also this context tree weighting algorithm. Again, he did a great job explaining this, so I don't need to go into the details. Next, there's permutation entropy, where you go through the signal, you look at these consecutive patterns, and you count or you measure the ordinal rankings of these triplets of points within these patterns, okay? So you basically have this mapping of your signal into these discrete symbols. So for instance, the first symbol would be high, middle, low, and the second signal, or the second symbol rather, would be low, middle, high, and so forth. You look at how these symbols are distributed, and the more evenly distributed they are, the higher the permutation entropy. And then lastly, you have some measures that are based on reoccurring states of the signal, okay? So that would include the sample entropy, and then also the multi-scale entropy, which takes into account the fact that the signal looks differently or behaves differently at different time scales. So you see a time series here, you see that there are different points colored differently, which reoccur within a certain tolerance, indicated by this dotted line, and that's the basis for sample entropy and multi-scale entropy. So we applied these measures to the same data that Katrin presented on earlier. So I don't need to go too much into the experiment. You're already familiar with the local global paradigm. Just to refresh, these were fetuses recorded as early as 25 weeks gestation, and then also newborns recorded up to 8 weeks after birth. So we had 101 MEG recordings in total, 43 fetuses with longitudinal data, 20 newborns, including 16 of whom were actually recorded from before birth. So that's really nice. We have some longitudinal data before and after birth. Okay? So as far as the data processing and so forth, we take these signals, we do a trial average. We actually take 3.2 seconds of data from this local global paradigm. So all the way from 200 milliseconds before the first tone to a full second after the final tone. We do it this way because we have a 1 to 10 hertz low pass filter for the fetal data and a 1 to 15 hertz low pass filter for the infant data. And if we just took 1 second of data, say after the final tone, we might at most have only 10 oscillatory cycles. So we wanted to take a lot of data, and we averaged not only across trials, but also across channels. The reason being that there's a lot of variability in the head orientation of the fetus, and even the newborn that's lying in this cradle, right? So if you were to compare the same channels in different recordings, that would actually be apples to oranges. They're the same channels, but the head is in a different position. So you see how there's these, like 5 to 10 channels that pick up the cortical signals, and we average across those channels. So again, it's just really important to emphasize, we don't have the spatial dimension, so it's not quite the SPCI, but it's inspired by perturbational complexity. Our hypothesis was fairly intuitive, I would say. We thought, okay, the thalamocortical system matures, it becomes more efficient at integrating information. So in the context of IIT, which Pedro mentioned, we thought, okay, yeah, the fetuses, they'll have a low capacity for consciousness, at least at the very beginning, at 25 weeks gestation, low entropy. Then, as they get closer to birth, they'll have higher entropy, higher capacity for consciousness. And then you'll have the newborns, they're already born, they have a more mature thalamocortical system, higher capacity for consciousness, higher entropy. Guess what? We were completely wrong, okay? So we had this very clear negative correlation between gestational age and entropy. Very unexpected. So this is the Limbal-Ziv complexity, and I show it separately for males and females because actually this looks different depending on how you break it down by fetal sex. Also very surprising. So of course on the x-axis you have the postmenstrual age, on the y-axis you have the entropy, the red data points are fetuses, the blue data points are newborns, dotted lines connect recordings from the same subjects. The magenta data points are the averages within these 1 week time bins, and then you have the least squares fit, right? So actually, once you do this smoothing, you see that the majority of the variance is explained by the postmenstrual age, both in males and in females. But it turns out, for all the measures we looked at, we see that there's more variance explained by postmenstrual age in males rather than females. So I know that's a lot of scatter plots to look at, right? But I'll just show you, these are different entropy measures, and we broke it down by sex as well, okay? So in the interest of time, I won't bore you with the statistical details, but you can just take my word for it. We had this main effect of gestational age, a sex by gestational age interaction, and even a main effect of sex in some of the entropy measures, okay? So this is actually consistent with some earlier work that was done in the other fetal MEG site in Little Rock, Arkansas. They did this study a few years ago. They saw that if you look at visually evoked responses in fetuses, you see that they're significantly harder to detect in female fetuses. Why is that? Well, the female fetuses actually have more complex signals, okay, at least according to our analysis. So if you think about a more complex signal, the complexity of that signal may mask this evoked response. Okay, so you think about higher complexity, it's higher to see this evoked response that's sort of hiding in there, right? So that fits with previous work. Another way to think about this is, look, early in development, you haven't had these processes like synaptic pruning, apoptosis that shape these circuits. So you have this very noisy time series. And then, circuits get sculpted by development, and these auditory evoked responses get larger and larger, and if you measure the entropy relative to some fixed threshold, it starts to decrease. So we tested that idea using this entropy decomposition, which Pedro had already developed and published on. And not to bore you too much with the details, but the idea is you take these signals, you scramble the Fourier phases and amplitudes, and this is a clever way of deducing whether the change in entropy is due to the phases or the amplitudes, okay? So we did this for all of our entropy measures, and what we found is that these decreases in entropy are actually due to changes in the amplitude. So that's what you see on the left in cyan, and then on the right in pink, we lumped the phase and the phase by amplitude interaction into this non-amplitude term. And we found that at least for permutation entropy, these changes in non-amplitude properties of the signal actually increase the entropy with maturation. So that could potentially be, consistent with the idea that entropy would increase with consciousness. It might just be masked by what we see as a result of the amplitude properties of the signal. So again, yeah, we found this in all of the entropy measures, and this was significant for almost all of them in fetuses and newborns. How do we explain what's going on? A little bit counterintuitive, right? So there's this idea of canalization, which basically says, okay, early in development, you have many different possible circuit configurations, yeah? Like things haven't really been constrained yet by development and genetics, so there are many different states that the system can take. And actually, the definition of entropy is a number of possible ways to arrange a system. So basically, by definition, you have higher entropy early in development. So that's what you see on the left. And then as you have synaptic pruning and apoptosis, it's as if the system is being squeezed through this narrow canal, right? So you come out this canal to the other side, and you have this circuit configuration that's been very constrained by genetics and synaptic pruning and so forth. And so, because this constrained circuit has few possible configurations, it has low entropy. All right, so that's one possible way to think about our results. So just to wrap up, what we see as a result of the amplitude changes in the signal probably has nothing to do with consciousness, but we saw this hint of an effect due to changes in signal phase maturation. Could be related to consciousness, we don't really know. I think it's important to emphasize this isn't quite SPCI yet, but we advocate for future studies that would have the spatial resolution to measure something that really is the SPCI. Big thank you to FET Open Luminous for funding this project, and to all my collaborators, most of whom are here at the symposium today, but also Julia Moser, who did so much work here as a PhD student in Tübingen. So she's in Minnesota, shout out to Julia, and I will take questions now. Yes.
Thank you very much. That's a really interesting talk. So I'm a small question about the measurement, because I see that this entropy measurement is actually strong. Depends on how complexity of the signals, and actually the how complexity of the signals is also very strong. Depends on the noise, right? So is it some kind of how do we measure the signals? Actually, the signal qualities in the very early development of the kids, and maybe this due to the very noise of the signals, and then it will be more complex, and then it will be very entropy. So is there some way we can isolate the effect and so say, okay, actually our measurement is very inverse against the noise?
Great point. You have more noise, you have higher entropy. One thing I would emphasize there is even just within the fetuses, we have this effect. And I don't think there's a reason, Katrin can maybe correct me if I'm wrong, but within the fetal recordings, the noise should be similar. So I don't think we would have this within fetuses if it were just an effect of noise. And Katrin has done some great work actually estimating the SNR in these signals. I can talk more about that at the end if there's time. I really need to give the stage now to Asger.